Have you ever wondered how microbiologists can identify bacteria within just a few minutes under the microscope? The answer lies in one of the most important staining techniques in microbiology, that is gram staining. So in today's video, we will learn the principle, reagents, procedure, interpretation, clinical importance and common errors all in a simple and easy to remember way. So gram staining first developed by the Danish scientist Hans Christian Gram in the year 1883. Interestingly, he developed this technique while studying lung tissue, but today it has become the most widely used staining technique in diagnostic microbiology. So the stain divides the bacteria into two major groups, that is gram-positive bacteria and gram-negative bacteria. So the stain is a type of differential staining. So we're going to know about the principle of gram stain. So it's based on mainly three principles. So first is the cell wall theory. So the gram-positive bacteria have thick peptidoglycan layer and low lipid content, and they have numerous teichoic acid cross-linkages. And the gram-negative cell walls, which consist mainly of thinner layer of peptidoglycan and an outer lipid bilayer that is dehydrated during decolorization. So the difference in the composition between the cell walls accounts for the gram staining differences. So what happens is, during the decolorization step, the alcohol dissolves the outer membrane of the gram-negative bacteria, allowing the crystal violet iodine complex to wash away. Whereas in gram-positive bacteria, this complex is retained because their thick peptidoglycan layer becomes dehydrated and traps the dye. Next theory is the pH theory. So in this theory, the cytoplasm of the gram-positive bacteria is more acidic, hence can retain the basic dye for longer time. Next comes the magnesium ribonucleotide theory. So in this theory, it states that it is present in the cell membrane of gram-positive and it helps them to retain the primary dye. So these are the four reagents of the gram staining. First is the grams crystal violet, second is the grams iodine, third is the grams decolorizer, and fourth is the safranin. So now we are going to see the procedure of gram staining. So first step is fixation. The smear made on a slide from a bacterial culture or specimen is air dried and then heat fixed. So first comes the step 1 is the primary stain. So the smear is stained with paras or aniline dyes such as crystal violet for 1 minute. Then the slide is rinsed in water, and so this lets the crystal violet stains all the bacteria violet in color. Next step is the mordant, which is the grams iodine. So grams iodine is poured over the slide for 1 minute. Then the slide is again rinsed with water, and in this step the grams iodine acts as a mordant and binds to the dye to form bigger di-iodine complexes in the cytoplasm. The third step is decolorization. So in there we pour few drops of decolorizer to the smear, for example acetone, and it is just kept for 2 seconds. And the slide is immediately rinsed with water. The decolorizer removes the primary stain from the gram-negative bacteria, while the gram-positive bacteria will retain the primary step. So decolorization here is the most crucial step of gram staining. If the decolorization is poured for more time, even the gram-positive bacteria can lose color and appear as gram-negative bacteria. So it is called over decolorization. And if poured for less time, the gram-negative bacteria do not lose the color of the primary stain properly, and it is known as under decolorization. The fourth and the final step is the counter staining, which is mainly done by safranin, and it is done for 30 to 60 seconds. So it imparts the bacteria pink or red to the gram-negative bacteria. Alternatively, we can use neutral red also as a counter stain, especially for gonococci. So the slide is rinsed with tap water and dried and then examined under the oil objective microscope. So next comes the interpretation. So the smear is observed in the oil immersion objective. So the gram-positive bacteria resist decolorization and retain the stain of primary stain, that is, it appears as violet color. And the gram-negative bacteria are decolorized and therefore they take up the counter stain, that is pink color. So for the gram-positive bacteria, the observation is violet-colored spherical-shaped organisms arranged predominantly in clusters, which are seen, and inference will be gram-positive cocci in clusters. And the examples are Staphylococcus aureus. So for the gram-negative bacteria, the observation is pink-colored rod-shaped organisms which are arranged discretely, and the inference will be gram-negative bacilli arranged singly. Examples will be Escherichia coli, Klebsiella pneumoniae, Shigella dysentery, Salmonella typhi, etc. So now here we are going to see the examples of various gram-positive and gram-negative bacteria. So the first is gram-positive cocci, which are mainly Staphylococcus, Streptococcus, and Enterococcus species. Second is gram-positive bacilli. Examples are Corynebacterium, Listeria, Lactobacillus, Bacillus, and Clostridium. Third is gram-negative cocci, that is Neisseria gonorrhea and Neisseria meningitis and Moraxella. Then comes the gram-negative bacilli, which includes Pseudomonas, all the enterobacterials, which has Escherichia coli, Klebsiella pneumonia, Proteus, uh, Shigella, Salmonella. Then there are certain other organisms also like Yersinia, Bordetella, Brucella, Vibrio, Campylobacter. So they are all gram-negative. So next we are going to see the uses of gram stain. So the first use is for identification. So the gram staining helps from bacterial culture, gives an idea to put the corresponding biochemical test for further identification of bacteria. Second is to start any empirical treatment with broad spectrum antibiotics. So gram stain from the specimen gives a preliminary clue about the bacteria present. So that the empirical treatment with broad spectrum antibiotics can be started early before the culture report is available. Third is the gram stain is useful for staining certain fungi like Candida species and Cryptococcus neoformans. Fourth is to identify the quality of the specimen. So the gram staining helps in screening the quality of the sputum specimen before processing it for culture. Presence of more pus cells and less epithelial cells indicates a good quality specimen. The fifth is anaerobic organisms such as Clostridium do not grow in routine culture. So therefore the organisms can be detected in gram stain, but aerobic culture negative gives a preliminary clue to perform an anaerobic culture of the specimen. So next we are going to see some common errors in the staining procedure. So sometimes if we put a low concentration of crystal violet, then there can be understaining and it can give faulty results. Next is if we are heating the slide more during fixation, it can lead to burning of the stain and it can even cause faulty staining. So third is excessive washing between steps. So it can lead to false positive or false negative results. The fourth is insufficient iodine exposure. The fifth is prolonged decolorization. And the sixth is excessive counter staining. And the seventh is preparation of the thick smear, which can lead to non-uniform staining. And so there can be a mixed growth seen. So this is a mind map which shows the overall procedure of gram staining as well as the differences in the gram positive and gram negative bacteria, which leads to the which is the main principle of gram staining. So that's gram staining explained from start to finish.
If you found this video helpful, don't forget to like, share, and subscribe the channel for more microbiology concepts, NEET PG division, laboratory techniques, and infectious disease updates. Comment below which microbiology topic would you like me to cover next.